Welcome to part one of putting it all together. So this is going to be about writing the results section. For an overview, we're going to go over what this might look for a quantitative paper, a qualitative paper, and discussing more about sort of what the resultant section entails. So congratulations, first of all, you've made it. So far, you've become acquainted with a new field of study, you've read sources and articles written for experts within that community, which is no small feat, and you've performed research and gained data. So what's next? Getting the research paper layout right is sometimes a little bit tricky and figuring out what goes where. So let's talk about that. The general layout for a research article will be the abstract, introduction, methods, results, discussion, and conclusions. For the purposes of this PowerPoint, you are here at results. The results section should be objective, clear, relevant, and concise. By objective, I mean you're reporting without bias. You're not analyzing the data yet. You might point out interesting trends or variances, but you are not going to talk about why you think those happened yet. It should be clear, so it should have a logical flow. It should be easy to follow. The findings should be easy to follow, and they should clearly link and clearly transition from one to the other. They should be relevant, which means don't include any data that isn't part of your story. A lot of research papers have a section called the supplemental section, and this is where we might put data that is interesting or tangentially relevant, or might make a point that is not huge to your story, but is still part of the research that you did. So consider moving things that are less relevant to that section of the paper or just including them entirely. Finally, it should be concise, which means don't bog it down with too much detail. So why would you write a results section? First, it removes bias by presenting your data without analysis and without a really personal input. It also explicitly addresses the hypotheses and if they were supported or rejected by the data. So this is important because this sets up really well for the discussion where you're going to go into more of the nuance, and that's where you'll get to sort of take that data that's been presented quite objectively and use it to tell the story that you are seeing with the data. It's also really important to allow other scientists to perform replication studies. This is where a scientist in a different lab might read a study and say, wow, I think that might be relevant to what I'm doing. That's so interesting. Or we hope not, but they might also say, I don't know if I actually believe that this happened. And they could take your methods and materials, reperform these studies, and hopefully they would replicate the findings, which will be represented in the results section. Let's go over the structure of the results section now. So you can structure it by hypothesis, which means you're going to really systematically go through and address your research questions as individual sections and whether or not at the end of those sections they were supported or rejected. You might also choose to represent them by theme, where you might group similar findings together, or you might choose to represent them in chronological order. I'm going to really caution you against chronological order. There might be situations in which this is useful, but especially in scientific writing, it is rarely appropriate to represent your data or to present your results strictly in the order you found them, because often that does not contribute to telling the story, and it makes the data, even relevant data, seem irrelevant. So be very careful with that. Another really important aspect of the results section is our visual aids. Visual aids are our graphs, our schematics, and so I wanted to go over what that might look like. So here I've pulled a figure from a paper by Dr. Marjan Rafat that was published in 2018 that was looking at triple-negative breast cancer recurrence after radiotherapy. I pulled this figure because I like the way that it combines different media. So first, she shows us this schematic, which she doesn't have to. She could have just described it in words, but it really does help to paint the picture of what's going on here, where we can see clearly there is a tumor sink here. This is where the radiation is occurring. In the context of the paper, we're finding that in mice, in mice under certain conditions, these tumor cells will migrate to the irradiated fat pad here. She also presents images of what she found by looking at the luminescence of the cancer cells in the fat pad, the muscle, and the peritoneum of the irradiated mice versus the control mice, which really paints the picture and really drives home the hypothesis that it is the radiation that's making this occur. And then she also represents her data as plots. And something I want to draw your attention to is how how the plots are labeled. So on the y-axis, we are looking at our luminescence levels from these cells, and they have a unit, which is photons per second. So the reading from this was photons per second. It's important that your y-axis is appropriately labeled, and we'll go more into that at a later time. Also is the way that the data is arranged. So the data is arranged by control and irradiated. Sometimes it might make more sense to put your irradiated memory fat pad next to your control fat pad, or in whatever context your data needs to be presented. But in this case, I think it was a smart decision to represent them separately because you can very clearly see how they all increase from the control. The next thing you should know about your visual aids is you should be able to set up your figure captions and in-text references. So I want to draw your attention to the caption from the figure we just looked at. In the caption for this panel, these panels E and F, you can see where it's discussed that there is a control and where there is and isn't statistical significance, as well as what statistical test was performed, what the p-value these stars represent is, and what the error bars are, and also if something is non-significant, what that means. It's very important to include these in the figure caption because it is part of what makes your figures reliable and believable. Another thing to notice, again, here in ENF, is the number of N. So N are also always indicated for each condition. And if you were to go and count these individual data points, 1, 2, 3, 4, 5, 6, 7, you will find 7N, just like she says in the figure caption. So be sure that you are representing your data appropriately in the figure caption. When you're doing in-text citations, or references rather, to your figure, you can either reference them by saying something like, as is seen in figure 1, but more frequently you will see scientists make a statement and then reference a figure like this. Another thing to notice here is that the figures are referenced in the order that they appear as panels in the figure. So we're not referencing figure 2E before figure 2A. This is best practice and is typically what's done. So visual aids are there to make it easier to understand your data, to make your trends more visible, and to simplify very complex data. Your visual aids are not there to show results with little in-text discussion or context. They are not sufficient to present your data. Your text should be the main presentation and description of your data, with the visual aids simply being an aid to increase understanding of your data. So let's look at what this might look like for quantitative and qualitative results. Quantitative results, this is going to be, you might present things like descriptive statistics, tests performed, and outcomes. For qualitative results, you might organize them by theme and use quotes or excerpts to highlight your results. So an example of a quantitative results sort of excerpt here might be: the analysis revealed participants who exercised for at least 30 minutes daily reported significantly higher well-being scores. They represent their mean and their standard deviation here, than those who did not. Again, mean and standard deviation. And then they represent their N as well as what their p-value will be, what their p-value indicates. For the qualitative section, for a very similar experiment focus, it might be something like: participants frequently mentioned the stress-reducing benefits of exercise, with one respondent stating, "My morning walk helps me clear my mind and focus better throughout the day." So we can see how different these are, but how each of the fields uses their strength and highlight to represent the results. All right, so let's check ourselves. When you're going through your results section, you should ask yourself, are my findings relevant? Are the results I'm presenting relevant to my hypothesis or research question? If it's kind of, but also more distracting than it is helpful, maybe it belongs in the supplemental. If it doesn't really tell anything about your story, but it's work that you did, unfortunately, the results section and this paper just might not be the place for it. Does the section flow logically? You want to make sure that as you are reading it, you are stepping from idea to idea, not flying all over the place, touching on this, then going back to that. I would recommend either reading it out loud to yourself or handing it to somebody who has not done this research with you, asking them to read it, and then asking, does that make sense? And if the answer is no, why? And use that feedback to help you formulate your results section better. Do the visual aids highlight and increase understanding? This is important because I have seen on multiple occasions visual aids that I feel actually didn't represent the data very well and gave a different uh, implication than the data actually showed. I've also seen figures that don't, to me, just confused me more and made the data harder to understand. So make sure that someone outside of your research team or outside of you understands, feels that these visual aids, tables, graphs, schematics help you to understand things better. Make sure you have enough of them as well. Sometimes it takes somebody else reading your writing to say, hey, actually, this doesn't make a lot of sense. And I think a schematic here or a drawing of what's happening would really help me to understand better what's going on. Are statistical values represented? Again, you want to have indications of significance both on the plots and then described in the captions. And in the text itself, you want to reference maybe important statistics like means, but it's also appropriate just to reference the visual aid or the figure that has that in the captions. And finally, that's for quantitative. That's for quantitative research, of course. For qualitative research, are these findings summarized and supported? So, thinking about that example, I don't think it would have been enough for them to say that people generally said that exercise was good for them. It was really strong and helped a lot that a quote from somebody who was interviewed for that study was provided. Um, to qualitative research, that is the same thing as presenting data points that you got in quantitative research.